Hi everyone, welcome to the Live Longer World podcast. I am your host, Astha Jain. On this podcast, I have conversations with scientists and entrepreneurs that are transforming the field of longevity science. If you wish to stay notified of upcoming podcast releases and sign up for my newsletter, head over to livelongerworld.com. My guest today is Dr. David Gems, who runs the Elegans Aging lab at University College London. We had a fascinating discussion talking about emerging paradigms in the theories of aging. We discussed the hyperfunction theory or the programmatic theory of aging, which gives a better integrated understanding of the different theories of aging that have been presented so far, such as damage accumulation, evolutionary theory of aging, and antagonistic pleiotropy. It's been assumed that molecular damage is always the cause of aging, but the programmatic theory of aging shows that that might not be the case. We also discussed David's critique of the hallmarks of aging and how there should be a better paradigm for the aging field. Lastly, we discussed how research from his lab on C. elegans might explain some of the conflict around whether sirtuins affect lifespan, and how C. elegans also show versions of this programmatic theory of aging, which can give us a better understanding of how to study aging going forward. Just a quick note, there is a point in the discussion where David pulls up some slides to better explain the programmatic theory of aging. So if you're listening to the audio-only version, you might want to refer to the YouTube version or the show notes. With that, I hope you enjoy my discussion with Dr. David Gems. Hi, David, and welcome to the Live Longer World podcast. So great to have you here today.
Great to be here.
Thanks for your time. So I want to start by talking about one of your recent reviews, which is the hyperfunction theory of aging, an emerging paradigm in the biology of aging. And I think it's a really interesting review because you talk about some of the different theories of aging, like evolutionary theory, antagonistic pleiotropy. You also talk about the programmatic theory, and then you say that we need one integrated understanding of the theory of aging. And maybe this hyperfunction theory or the quasi-program theory is one such theory. So maybe if you can discuss briefly what some of the other theories of aging are that have been presented before, and what is this emerging paradigm which has been called the hyperfunction theory?
I mean, I suppose that most fields of science that are at a state of relative maturity, like something like, say, chemistry, they have kind of foundational ideas which unite the field and about which there's very little argument, and which really, in a way, are what makes the field so powerful. So, for example, in chemistry, you've got something like the periodic table and the understanding of the chemical bond and atomic structure and so on. And all chemists agree about that. The chemists don't argue about whether the periodic table is real or not. And the thing about these kinds of foundations is that they really guide guide the research fruitfully and usefully. So they produce predictions and hypotheses that have a good chance of being right. And the thing about the aging field is that, in a way, that sort of paradigm, that kind of general paradigm, those foundational ideas don't really exist for aging, not in the same way as in more mature fields. So the article about the hyperfunction theory, I mean, it's looking at actually several new theories, which I'm not arguing that these are necessarily absolutely right, but that they look like that. I guess it's arguing that there should be such an understanding because there's only one nature, there's only one reality. So why shouldn't it be possible to understand aging in a general way? And I think that having worked in the field for many decades, these new ideas they seem very they present clearer sort of understanding of aging, certainly than I've ever had before. So I think one could characterize the kind of aging field at the moment as like a collection of different theories and ideas. which are not really integrated in any major way. But I think in the past, I think especially during the 90s, there was a collection of ideas about aging that seemed to kind of gel together in a fairly coherent way. And that included, for example, the evolutionary theory of aging, which I think remains, valid and undoubtedly correct, which is sort of about the why aging evolves, why it's there. But then the sort of the more difficult aspect of aging is really what are the mechanisms, the actual, what actually happens during aging that causes aging-related disease and causes death. And during the 90s particularly, I think there was a consensus that emerged in the field that that aging is very much a process of kind of stochastic breakdown. So rather in a similar way to complex machinery or buildings and so on, that inevitably things wear out and break down over time. And I think probably influenced by the sort of fashion for explanations at the molecular level, that very much led to an interest in the idea of molecular damage accumulation. So, for example, DNA damage to protein and damage to lipids and so on. And ideas about the importance of metabolism in aging, which kind of go way back to the rate of living theory, back from the 1930s, led to a kind of convergence on the idea of metabolism and oxidation as a sort of driver of aging. And certainly when I came into the field, my impression from the field was that it had been established at that point. It was now beyond any shadow of a doubt that the main cause of aging was actually oxidation, was oxidative damage, particularly from reactive oxygen species such as superoxide free radical produced as a byproduct of mitochondrial metabolism. So I think that a number of ideas kind of got linked together in a very in a rather elegant way that looked like very much like a sort of periodic table for aging. So you had, for example, the idea of metabolism generating oxidative damage, reactive oxygen and damage accumulating, and you had the idea of the disposable soma, which was a beautiful and elegant a kind of connection between the evolutionary theory and the idea of damage accumulation. And the reasoning was that the processes of maintenance that protect against damage are quite resource costly. And in the wild, organisms, they don't have all the resources, unlimited resources, and they may have to sort of decide how to invest the limited resources they've got to optimize fitness. And actually, it doesn't make sense to invest so well into maintenance that you actually don't age. You're actually better off kind of maintaining yourself just well enough to be able to reproduce, and that way you can actually divert resources into reproduction, and that way you out-reproduce other organisms in your species, and so it favors the evolution of a disposable, right, short-lived soma, rather like, I think, disposable consumer products sometimes, produced. They're produced to be, they're made to be able to produce in large numbers rather than to last.
Mm-hmm.
So this was a beautiful connection of the evolutionary theory and the mechanistic theory. And then there was the dietary restriction, the allocation theory, which was an idea that you could understand dietary restriction because what dietary restriction did was to shift resources away from reproduction and into maintenance. So it all kind of linked together very elegantly. But I think, I'm very influenced by experimental work. And I mean, my lab, we've been working with C. elegans, trying to test different theories of aging. And one of the things that we did was really to try to test the oxidative damage theory in C. elegans, which in the early noughties was still very much sort of the mainstream. And it was actually kind of almost a rather an eccentric thing to suggest that there might be a problem with that theory in the early. But the results that we were getting for C. elegans were really just repeatedly not supporting the theory. We did a whole series of very sort of tedious papers where we would test the theory and then the theory wasn't supported. So we'd say, well, it doesn't support the theory. And there was always, well, yeah, but maybe you haven't covered, there's always this possibility. So you could never completely disprove this theory. Whatever you did, the data generally didn't fit, but you couldn't disprove it. So I think certainly by about 2008, 2009, there was kind of a shift in the field. It was really quite sudden, where before that, at aging meetings, there were lots of talks about oxidative damage. And then after that, very few, certainly in the English-speaking world. So there was a sort of, I guess, what you call a watershed in the field, where, in a sense, the sort of the grip of belief in that theory kind of suddenly dissipated. But that kind of left, I think, a question about really the whole idea of damage as a cause of aging, what I would call what I call sometimes the damage maintenance paradigm, which is in a way the kind of the closest thing to a unifying paradigm for people studying mechanistic biogerontology. So I think in a way what's interesting about the programmatic theories is that they, unlike the oxidative damage theory or disposable soma, these theories are much more grounded in experimental work over the last 20 years. Whereas, I mean, the Ross theory came out of just a kind of armchair speculations from Denham Harman, and the disposable somas similarly was just a kind of a, piece of conjecture really. It wasn't something that kind of arose in a way to try to explain a lot of existing evidence. So I think in a way the emerging theory, including the programmatic theory, which I think is really just part of the bigger picture, in a way makes much better sense of a lot of the findings over the last 20 years from biogerontology.
Yeah, no, that's super interesting that you explain the history. It's like, just the way science progresses. And you've also spoken about some of this in your critique of the hallmarks of aging, how it's just assumed that all of aging is a direct or indirect cause of molecular damage, which might not be the case. And we can talk about that later, but maybe if you can explain, what is this hyperfunction theory or the programmatic theory?
So, I mean, I would first of all say that I think the programmatic theory doesn't, it's got different names. I mean, it's Blagascloni, Misha Blagascloni is one of the people that kind of has developed the theory, I think one of the originators of the theory. And I kind of started calling it the hyperfunction theory, so he now does. But then there were, at the same time, João Pedro de Magalhães proposed a very similar theory, which has been called the developmental theory, which is, as I say, they're, I think, really, in many ways, the same theory. And I call it the programmatic theory for different reasons. I think it's a little bit more precise. Yeah, how to explain the theory? So, one of the things that came out from the genetics of aging, so this is looking at mutations that cause increases in lifespan, was the finding that pathways that are involved with growth are very important for aging. So, for example, in DAF2, the insulin IGF signaling pathway, if you knock it down, you can double, triple, quadruple the lifespan of C. elegans. And one of the things that we did here at UCL early on was to test what happens if you knock the pathway down in fruit flies and you see increases in lifespan. So, and similar work in mice, particularly actually work from André Bartke in the States, showed that knockdown of this pathway is also extending lifespan. So it seems to be a universal effect on aging of this pathway, which is involved in growth. So this was a big puzzle around 2000. You have this growth pathway, which is having effects on aging. One of the things that was confusing at the beginning was the fact that it seemed like you could separate the effects of the pathway on body size from the effects on lifespan. So that led to the idea that the effects on growth were probably nothing to do with the effects on aging. And also the other assumption was, well, we have to somehow understand how this pathway is affecting damage and maintenance, because that's what aging is. Aging is damage and maintenance. So it must be somehow affecting damage and maintenance. And in fact, the pathways do have some effects on damage and maintenance. So part of the growth control pathway, for example, the targeted rapamycin pathway, regulates levels of autophagy. And certainly, for example, in C. elegans, if you the long-lived mutants, they're quite resistant to oxidative stress and thermal injury and so on. So it seemed to fit that actually the pathway was acting on aging through effects on somatic maintenance. But attempts to test the damage maintenance theory were coming up negative, so it was really confusing. What the Blagasconi and de Magalhães argued was that somehow it was growth that was driving aging. So somehow it wasn't damage and maintenance. This was something that both of them sort of, in a way, conjectured. They said, look, the damage maintenance theory seems inadequate. And that's an important sort of, it's like a, let's put the damage maintenance theory, just for now, let's put it to one side. And let's look at whether it's a growth effect. So how could that work? One of the problems was the fact that the implication was that there's some sort of developmental type program for aging. And this immediately kind of flashed a red light to people in the aging field, especially those familiar with the evolutionary theory, because it suggested the idea that you're saying that aging is programmed. And one of the kind of axioms of the evolutionary theory is that aging is not programmed. Aging is not an adaptation of any kind. It's like a byproduct of the way that natural selection works, favoring traits in early life. But then what Blagasconi pointed out is something I was very conscious of this as well. That the word program is complex. It's problematic because, like the word aging, it has multiple meanings, right? So programmed can mean programmed in the adaptive sense. So it means that it's a program for something. So to say that there's a developmental sort of program for aging would imply that aging is adaptive, but that's wrong. So it can't be programmed. But then program can also mean programmed in the mechanistic sense, right? So there are mechanistic processes involved. So for example, I don't know, in cancer, for example, cancer cells, they're undergoing the cell cycle. So that's the cell cycle program which is being enacted. But cancer is not an adaptation. So it's so you've got something that's mechanistically programmed, but not kind of programmed in the sense of being an adaptation. So what Misha said was, Blagasconi said, well, okay, it's programmed, it's not programmed, it's quasi-programmed or quasi-programmed, I think, if you're in America, right? So it's sort of so then that produces a new word, which is the quasi-program. And so what Blagasconi was arguing was that actually what you get in late life is essentially quasi-programs where programs that have evolved a function in early life, they run on in later life in a way that is essentially futile. So you get an adaptive program runs on to become a pathogenic quasi-program. And in fact, actually, this kind of picture is very similar to something that George Williams said back in whenever it was, 57. George Williams put forward the idea of antagonistic pleiotropy, which is really I think the most plausible version of the evolutionary theory of aging, where genes essentially are having beneficial effects in early life and detrimental effects later. And I mean, the big problem and the big challenge, I think, has been to understand how that actually happens in terms of mechanisms, right? I think if you solve that, you probably, in a sense, you've really got to the bottom of things, with aging, because aging is largely genetically determined. Actually, what Williams said, Williams had this one example that he gave. He didn't think a lot about proximate mechanisms of aging, and he imagined this is kind of fantasy, really. He imagined a gene which is involved in deposition of calcium, And so this gene, you've got a new variant of the gene, which increases calcium deposition into bone. And so what that does, it speeds up bone growth. And if you imagine something like a sort of a gazelle, you know, in Africa, and so the bones grow faster, and then when the cheetah comes, it can run faster, it can escape, so it increases fitness. But the same gene, this calcium deposition gene, is actually, in later life, is causing calcium to go into vascular tissue. It's causing vascular calcification. It's promoting arteriosclerosis and disease. So here, this is actually very similar to what Blagoscloni is saying, because what you have is a function which then becomes essentially, it runs on and becomes pathogenic. So you have a program for bone growth becomes a quasi-program for arteriosclerosis. The difference is that in Blagoscloni's scenario, you're looking at entire developmental programs rather than individual biochemical functions. And that it provides a really elegant explanation for why these growth control pathways like the insulin-IGF signaling pathway in mammals, that's particularly IGF-1 signaling, and things like the targeted rapamycin, which is fundamental to cell growth, why they're controlling so many different diseases, because they're involved fundamentally in development. All development requires, right? It requires TOR for cell growth, but then in later life, the TOR is involved in the development of senescent pathology. So what's happening in the long-lived mutants is that you're dampening down the growth pathways, you're dampening the development, you're delaying the development of these quasi-programs, or you're blocking the quasi-programs, you're delaying the development of senescent pathologies. And in that's how you're seeing the effect on lifespan.
That's fascinating. Yeah.
It's a beautiful theory. And this is why I use the word programmatic, because what it's the point about programmatic is that calling it developmental theory is a little bit confusing, because it implies that it's actually ontogenesis somehow which is causing aging. But actually a lot of the changes we're talking about here are they're sort of developmental changes, but they're in things like tissue remodeling, tissue homeostasis, reproduction, immunity, and all of these things, complex programs involving cellular change, complex changes in gene expression, and so on, right? So they're not really developmental in the sense of ontogenesis, but they are programmatic, right? And programmatic also points to the ambiguity in the word program. So it's like, it's not programmed aging, it's programmatic, right? So that's acknowledging this issue. So, and in this scenario, damage isn't really playing much of a role at all. This is actually your wild type genes are driving the development of pathology. And actually what Misha argues is that the damage that you observe, you see molecular damage accumulating in late life. It's a consequence of the pathologies that are developing. It's not the cause, right? So it's a correlation, if you like. As he himself said, during my studies of aging, I've accumulated a lot of knowledge of the Ross theory. Does this mean that this causes my aging? Not. So that's the sort of that's the gist of the programmatic theory. So it's really, in many ways, it's kind of an alternative to the disposable soma theory. Because the disposable soma theory, what's so great about disposable soma theory, it's like a model for an ultimate proximate theory. And I guess when I came across disposable soma, I was very excited, and I thought that is what an explanation of aging looks like. You know, it's an ultimate proximate explanation. But I think it's probably just not correct, unfortunately, because it's based on the damage maintenance paradigm, which isn't correct. It's not that damage maintenance doesn't play any role. It probably does. And I can't emphasize enough that the programmatic theory, and Blagoscloni emphasized this particularly, it's not the whole of aging. It's really like it's like a major part of aging, but in itself, it's not sufficient to explain aging.
Interesting. You actually, in fact, also show this in through C. elegans research, and you have a preprint that shows that reproductive death in C. elegans, it was viewed as distinct from aging, but it might in fact be exaggerated versions of this programmatic theory. And then there's another one that says that C. elegans eat up their own intestines, which also might be a version of this wild type biological gene runoff. So maybe if you can explain some of those papers, because I think there would be good examples to really explain what you're talking about.
What's so funny about it, that honestly, I can't emphasize enough that, these kind of emerging understanding that I feel like it sounds a bit crazy, perhaps, but I feel like I understand aging far better than I did 5 years ago, and I think a lot of that has been thanks to these ideas from Blagosconi and Dumagules, and actually also a guy called Vladimir Dillman, who actually, in fact, was Misha Blagosconi's father, who also published quite a lot on programmatic theories and actually a wider multifactorial model, which I think is a fantastic model. But I was led to all of this by studying C. elegans, by doing experiments on C. elegans. So it really kind of tore my mind away from damage maintenance, which I believed in for years, like everybody else in the field, or most people. But the odd thing is, though, that C. elegans in many ways is really different to a typical animal in terms of the way it ages. I think C. elegans is kind of weird and different in many ways, but it is sort of part of the same spectrum of biology, so that you can sort of recognize in the bizarre version in C. elegans the same rules that are actually operative in higher animals. So there's a bunch of problems. So it really comes to, this is a bit depressing, I think, for me in a way to talk about. But the thing that got me into working on C. elegans was the discovery of these long-lived mutants. I mean, it's a flabbergasting finding that you can get single gene mutations that can double and triple and quadruple lifespan. I mean, the implications of that are jaw-dropping, because aging is like, think of human aging. It's so complex. Even any individual disease, if you look at the literature on osteoarthritis, it's incredibly complex. All these different diseases, and so many of them untreatable, Alzheimer's, COPD, and so on. But in the worm, you can intervene upstream of all of it. So you can get to the aging process itself. So all the diseases of aging, whatever the worms get, you can actually prevent them all. It's mind-boggling. So you've got the possibility that you can intervene in aging in humans in some fundamental way. And, let's forget cancer research or cardiovascular disease research. You go for aging itself. That's going to be much better. I mean, that's what the worm work implies, and it seems crazy, but there they are. They're living, when you look at them, it's astounding to see the long-lived worms, when the wild types have died of old age. And it's in C. elegans, which is a sort of notoriously simple organism, but should be straightforward to understand, discover the sort of secret of aging in worms. So the new work that has come out is a bit sort of, in a way, undercuts it all in a slightly depressing way. So what we found is that if you look at the pathology of aging in C. elegans, it shows really extreme destructive changes to organs which are not typical of higher animals. So the largest somatic organ inside the worm is the intestine. It's a huge organ, relatively speaking, and it shrivels away, and it's like, most of the volume is gone within about a week of adulthood. And a number of like the gonad, part of the gonad just fragments into pieces. It actually disintegrates, and then you get these huge uterine tumors. So these sort of things don't normally happen in aging, but they do happen in certain types of organisms, which are ones that undergo reproductive death. So this is what's sometimes called semal parity in biology. So these are semal parous organisms that have a single sort of suicidal burst of reproduction. But one of the most famous examples would be the Pacific salmon. You know, they swim upriver, they make this enormous effort, and then they spawn, and then they just drop dead. You know, like within weeks of spawning. So what we thought that, you know, it's really odd the way that the pathology, it looks like a semal-parous organism, but at the same time, there's no obvious benefit to it. So it can't be semal-parous. You know, all this self-destruction seems to be quasi-programmed. It seems to be futile. And one of the things that happens is that the worms fill up with yolk. The whole worm is absolutely full of yolk after reproduction. And what we found a while ago was that the worms actually convert the biomass of the intestine into yolk. But the whole thing seemed pointless in terms of it didn't seem to be anything to do with fitness. But then a graduate student in my lab called Karina Ken discovered that actually the yolk is actually vented out through the worm's vulva in large quantities. And in fact, it can support larval growth. So the implication is that in fact, it's a form of lactation. We call it yolk milk. So the worms are basically, they can't reproduce anymore. So they essentially convert their biomass into a ready energy source, and then they pump it out through their vulva to be to support the growth of the progeny. So one of the things in the preprint that you mentioned, if you take semal-parous organisms, so for example, if you take a salmon, you've got a salmon that's going to die in 2 weeks. Okay, so if you take Pacific salmon and you gonadectomize them, so you basically castrate them, you take away the gonads, so they basically don't undergo the reproductive death. You basically block reproduction. What happens then? Instead of living for a few weeks, they live 2, 3, 4, 5 years, because you've essentially got a suicide program. And you've blocked it. So, of course, you get a huge increase in lifespan. It's lovely work from an American biologist in California. It's from many years ago, Robertson. So we wondered if maybe that's true of C. elegans. In fact, so one of the things we did was to just look across the literature at what happens if you remove the gonad or you block reproduction what happens to lifespan. If you look in semal-parous organisms, you get huge increases in lifespan, like massive in some cases. But in the other type of organisms, so-called heteroparous species, which reproduce multiple times, if you prevent reproduction in them, if for example, through ovarectomy or gonadectomy and so on, in general, the effects are either there's no effect, or where there are effects, they're fairly modest. And so then what we did, we realized that in Caenorhabditis, you get some worms, some species that show reproductive death and some that don't. So if you look at the pathologies, there are some that kind of melt their organs and lactate and put out milk, and other ones that don't. The organs look fine. So if you gonadectomize the ones that melt themselves, so you have to actually remove the germline, and you do it with laser microsurgery, you get big extensions in lifespan. So we looked at various Caenorhabditis species, Caenorhabditis briggsii and another one called Caenorhabditis tropicalis that also melts its insides and does lactation. And then we looked at other Caenorhabditis species which don't, and then we took away their germline, and we see only very, very small increases in lifespan. So what we suspect is that the insulin and IGF signaling pathway and the germline signaling pathway is actually essentially promoting reproductive death. So what these big increases in lifespan, to some extent at least, it's similar to castrating a Pacific salmon. So that you won't see that. It's a biology which is peculiar to semal-parous organisms. But on the other hand, I think that's the bad news. So because it's sad, because it means that this miraculous increases in lifespan that you see in C. elegans, you're probably not going to translate that to humans, right? Because they're semal-parous and we're not.
I see. Because you're saying that's specific to their biology.
Yeah, it's specific to the semal-parous organisms. There's a whole other aspect of the story, which perhaps I won't go into, which is something called adaptive death, which can co-evolve with reproductive death. and probably has done in C. elegans and probably also in Pacific salmon. That's in a paper that we published last year called Death Happy. It's in the Proceedings of the Royal Society. But the good news is that you can make a very strong case that the sorts of mechanisms that are leading to death, in reproductive death, they are present in Itroparis species, but they're much more modest, right? And they are also highly programmatic, right? So in a way, I used to say that the C. elegans is like a sort of kind of massively Blagosclonian organism. It's like highly programmatic death in C. elegans. Molecular damage, for example, in humans, clearly does play a more important role. I mean, in cancer, for example. But so I think the working hypothesis is that sort of instead of the traditional view that you have semelparous organisms and then you've got Itroparis organisms completely different, and one of them is aging and the other one is sort of something totally different. Actually, the semelparous aging is like a greatly exaggerated version of mechanisms of aging that are operative in Itroparis species. So that means that aging in salmon is actually relevant mechanistically to mechanisms of aging in higher animals. It also means actually that plant senescence, like leaf senescence, these are other types of programmatic aging, that they are also relevant so to understanding aging in Itroparis species. So I think it sort of means that because plant senescence people generally, they're not invited to aging meetings, But I think they should be, because it's all part of the same spectrum of biology. So leaf senescence, very informative. I think leaf senescence and C. elegans aging, they share some things in common. This destructive biomass repurposing. So I worry my answers are probably a bit long-winded.
No, this is a good explanation. I was going to ask you. I mean, you said that the hyperfunction theory has sort of changed how you view aging, and you think you better understand aging now. I'm curious, how do you think this changes the dynamic of how we view aging in humans or just study aging going forward?
Well, I mean, my own view right now, and it's difficult because the field is moving so quickly. I find that, I'm sure that if I look at this video in a couple of years, I'll sort of think how wrong I was. But I mean, the thing that's in the recent essay on that I've called the hyperfunction theory, I think, which is really in a way more significant, is when you put the programmatic theory into the context of this earlier theory from Vladimir Dillman. So Dillman was he was a clinician and a biogerontologist who worked for many years in the USSR in Leningrad. I think he's probably the smartest and most original of the Russian biogerontology. He's sort of like the for me, he's like the Russian equivalent of someone like Tuck Finch or Alex Comfort in the UK, a sort of deep thinker. So he came up with a theory which, to me, looks tremendous in terms of possibly providing sort of the equivalent of the sort of periodic table in chemistry for aging. I mean, in the lab at the moment, I mean, we've sort of shifted a bit to doing a little bit less work on worms, to trying to use conceptual research to try to see if we can try to make sense of diseases of aging, to try to kind of put some provide a sort of perspective on the various competing ideas that exist for many diseases of aging at the moment, which I think a lot of the research on aging-related diseases is just as confused as the aging field itself. It's similar, that there's sort of like a swarm of different competing theories, sometimes that don't really connect with each other. So I think really the thing that interests me, I think in a way, the thing that got me excited in the first place was this idea that you could intervene in aging as a whole and produce the most sort of extraordinary effects. And in a way, the discovery of reproductive death and adaptive death in C. elegans is kind of, it's a bit sad really as far as that goes. It's kind of in a way, it kind of explains away the magic and mystery. For me, I don't know. Let's see whether other people are convinced by the work, you know.
Yeah.
But on the other hand, the emerging, you know, the programmatic theory, in a way, is part of a multifactorial model. As part of a multifactorial model, in a way, it provides a kind of, in a way, that a big missing part of the understanding of aging-related diseases. I mean, I could talk a little bit about the Dillman kind of multifactorial model, if you like.
Go for it. Yeah.
All right. So, should we try looking at the slides? So here's a sort of a really simple version of this multifactorial model, and that's why I like it. I don't like complicated theories. You know, this is really simple. So the multifactorial model of aging involves looking at causes of disease at a very, very high level of generality. Okay, so if we think about what causes disease in a very general way, and let's forget aging for now. Think about the diseases that mainly affect younger people, let's say under the age of 40. What is disease? Why does disease happen? So I think one could sort of define a kind of a very general paradigm about disease, which is that disease happens, and this is sort of from a biological perspective, when you have a normal function which is disrupted by something. And so then you have a dysfunction, a biological dysfunction, and that leads to disease. So, for example, things that cause, that disrupt normal function would include infectious pathogens, like viruses, like COVID, or like bacterial pathogens, or protozoan pathogens like malaria, macroscopic pathogens like helminths, and so on. So you've got all of that. And then you've got genetic disruption. So you have inherited genetic mutations or somatic mutations that's involved in cancer development, for example. And then you have all various other things like mechanical injury, dietary, you know, malnutrition, toxins in the environment, you know, bad air, smoking, whatever. So that all of these things are disrupting normal function. Okay. So, and I think that is a kind of that's guiding the thought processes of people involved in medical research in a very fundamental way. And I think the reason why aging has been so difficult, one of the reasons, there are many reasons actually, but particularly because aging involves something which is different to this. How do I go to the next slide? Right. So aging in a way has two elements. And this is a very, very simplified version of this multifactorial model. So you have disruption. So aging-related diseases, I've got here senescent pathologies. They are famously multifactorial in origin. So something like osteoarthritis, there are multiple things that can actually contribute to osteoarthritis. And that includes disruption of normal function. But these types of disruption of normal function, they're not the central aging process. They and what they impact is not so much the maximum lifespan of a species as the average lifespan, the mean lifespan. So the differences between individuals will be affected by the level of disruptions. If they, I don't know, they smoke or they drink to excess, or they overeat or whatever, or they're living in a place with very bad air or whatever. But actually, the main cause of aging is not disruption at all. The main cause arises from the wild-type genotype, the wild-type phenotype. What Dillman called, he called them normal diseases. So they're not arising from any kind of abnormality. They're arising from normal function. And these are the mechanisms which are predominantly programmatic in nature. Right, they're predominantly programmatic, and there are various different sorts of programmatic mechanisms. That's too much to go into, but I've got a number of essays in the pipeline about the different forms of programmatic aging. I think there are parts of the puzzle coming out, and so really your own wild-type genotype is killing you, it's destroying you. And the reason why it does that is because of evolutionary biology. And, you know, this is where antagonistic pleiotropy comes in. Essentially, our wild-type genotype is absolutely riddled with antagonistic pleiotropy. And that happens because of the declining force of natural selection with increasing age, what's called the selection shadow. But also, I would argue, and this is unpublished, a major factor here is biological constraint. And it's biological constraint, which is why you can't maximize all your fitness traits at once. And I think the key to understanding where diseases of aging are coming from is actually biological constraint.
Oh, sorry, what do you mean by biological constraint here?
Well, biological constraint means that biological processes are highly interrelated in such a way, they're highly interconnected in such a way that it's not possible to maximize benefits, all benefits at once. You have to choose, and therefore you have to make trade-offs. I mean, a very, very simple example, to use a very simple example. If you think about something like the synthesis of ATP, so if you increase, and this is in work from bacteriology, interventions that increase the ATP production rate will actually reduce the yield, and vice versa. So you can't maximize yield and rate at the same time. You have to basically come up with an optimal, right? You have to come up with a balance, a trade-off between the two. And if you have a mutation which actually increases the ATP production rate, it will show antagonistic pleiotropy, right? Because that mutation will also cause a reduction in yield. Right. So really, the argument is that really it's the biological constraint is at the heart of why antagonistic pleiotropy exists, and this is affecting many, many different traits. So according to this view here, then, in a sense, the programmatic aspect is what's been missed. You know, I mean, if you look at accounts of osteoarthritis, for example, which I've been looking at recently, most of them focus on disruption. They're always looking for disruption as an explanation, you know, and what the disruption will never explain is why a human being only will get osteoarthritis when they get into their 60s and 70s, whereas a mouse will start to get osteoarthritis after only 18 months. Right? And that's linked to the underlying biology of aging, which is programmatic. I don't know if I'll go to the next slide. Maybe it's too complex. Yeah, I won't. It's a bit small anyway. So this is all in this essay that you mentioned, the one in its Aging Research Reviews. It sets out the whole programmatic, the multifactorial theory, which is it's a sort of amalgamation of Dillman's thinking, which in many ways prefigured the programmatic theory, but wasn't as well informed because we know so much more information now about the pathways controlling aging and the links to growth and so on. But you can essentially kind of update Dillman's theory in the light of the programmatic theory. That's sort of the programmatic mechanism. So those are the folks that really, where the programmatic theory comes from Williams really originally, and then from Micha and Jean Pedro more recently.
Yeah, no, I actually think this slide where it's laid out so simply that, disease is just disruption of normal function, but then you have aging, which is not just caused by disruption of normal function. It's this programmatic theory that you just spoke about, also linked with antagonistic pleiotropy and biological constraint. And I think when you just look at this, look at it in terms of these two boxes, it makes a lot more sense. Just all that you explained right now.
I'm glad, and I find it's very weird. I find that, I mean, I've been looking a lot at osteoarthritis, and it's having this framework. It doesn't sort of provide all the answers, but in a strange way, it puts everything into perspective. It's almost like you sort of know where you are. You know where to put all the different findings. You can put them into a similar scheme, you know, and work out. It kind of also focuses attention at what the major questions are, which I think for osteoarthritis would be how constraint is leading to the age changes in chondrocyte function, which leads to these morphogenetic developmental changes in joints, which are really the major disease of osteoarthritis.
Absolutely. Yeah, it's nice to have better explanations. And I'll be on the lookout for some of your other papers that defines or talks about the different types of programmatic theory that you mentioned. But maybe I'll jump to another paper and I can remove this.
Yeah.
I'll jump to another paper where you talk about the FLN2 mutation, if that's how I pronounce it, that affects lifespan in C. elegans. And I thought this was really interesting because you show that, there's all this confounding research on whether sirtuin overexpression affects lifespan or not.
Yeah.
Some labs have been able to reproduce it, some others haven't. And now you show this new research that says that, well, it actually might be a mutation in a certain type of C. elegans that's affecting lifespan, and it's been falsely attributed to sirtuins. So yeah, maybe if you could just explain what you found out, what this mutation is, and whether and how sirtuins play a role there.
I mean, I think that in a way, this that most of the FLN2 story is like it's totally of no interest to anyone except worm people, because it's really this is to do with model organisms, and the fact that one of the sort of things that is so nice about C. elegans is that all the labs are using the same wild type, and we're all supposedly we try to use the same culture conditions. It's more or less the same. There are certain things you can't control, and what the paper showed was that actually there are two different C. elegans stocks that are being distributed, and one of them just has a mutation in it, which affects it. It alters it alters the way that the C. elegans die in a quite subtle way. But if you look at a number of publications in the past, you could see how the results had been confused and confounded by the presence of this mutation. So it's really just a sort of very technical paper for worm people to say, you need to always make sure that you don't have this FLN2 mutation. But the sirtuins thing is in a way of wider interest. So, there's a sort of interesting kind of philosophical, not philosophical, but there are questions about how you use model organisms, I think, which are really interesting here. So it's a bit hard to explain. Let me remember how the whole story went. So what happened was that it was reported that if you overexpress sirtuins, you get an increase in lifespan in C. elegans. It was one of the sirtuin variants called Sir 2.1. So what we found was that that if we backcrossed the strain that was distributed by the Senior Genetics Center with the long-lived strain. So we got the long-lived strain, we tested it, it was long-lived. But when we backcrossed it, the longevity went away. So we said, it's not actually it's not this 2 that's causing the longevity, it's something else. And we identified a second site mutation in the background. And then the original lab that did the work, they went and they checked and they said, yes, there is a second site mutation. But then the outcross strain, when we looked at it, it wasn't long-lived. But they said, our outcross strain is a bit long-lived. It's not as long-lived as before, but there is a significant increase. And that paper came out at the same time as our paper. And I just thought, what is going on? Are they making it up? I don't think so. And I spoke to the author of the paper, and I thought, there's no way that this guy made this up. I would not believe that. So in the end, it was just a mystery. So then it turned out that we had done the outcrossing with the strain with the FLN2 mutation in it. So when we found the FLN2 mutation, the very excellent postdoc, Yuan Zhao, who did that work, she said, oh, I'd like to look at the Sirtuin strains and see if it had any effect there. And I said, oh, no, not that again. But I said, all right, we should do it. You're right, we should test it. So she tested it, and what she found, she had the strain with the FLIN2 and without the FLIN2, and there was the sirtuin overexpression still didn't increase lifespan. So we thought, okay, good, it's not FLIN2. But then Yuan, being very rigorous cast of mind, she said, yes, but they used FUDR in their study. So, and we didn't. So we should retest our 2 outcross strains with FUDR. So FUDR, this is short for fluorodeoxyuridine or floxuridine. So people use it when they're doing lifespan experiments. They put it on the plates, and what it does is that it prevents the eggs from hatching. It basically blocks DNA replication. And more recently, a lot of labs avoid using FUDR because it's been found to cause artifacts. So what Yuan found was that when she used FUDR with the non-FLIN2 strain, that the sirtuin overexpression did increase lifespan a little bit. And the way that it did it was by reducing the infection in the pharynx, which affects about 40% of the C. elegans worms. And this is something that Yuan showed before, that there's 2 different kinds of death. So, for example, if you then do the same experiment, if you look in the presence of carbonicillin, which prevents all infection, then the sirtuin overexpression doesn't increase lifespan. So what that showed, and she was able to show that there was an effect on the invasion of the bacteria into the pharynx, where she could show that this was happening when you combined sirtuin overexpression with FUDR. You had to have FUDR to see it. So so we could reproduce the results not from the original Nature paper, but the paper that was published, that was Viswanathan and Grunty. We were able to reproduce it. So how do you interpret that exactly? I mean, we were very happy because I was always left with a bad conscience. Things never made sense, and now it made sense. So we argued about this. One argument which people made was, well, the effects of Sirtuin overexpression is an artifact of the use of FUDR, and therefore the information is not informative. But I don't agree with that, because I think really the question is, if whatever the sirtuin is doing, the overexpression is doing, if that mechanism the FUDR could be revealing a mechanism which is relevant to aging in other places. And that's really what matters. It's in a way, it's not about culturing everything correctly. It's whether the mechanism that you see is relevant. So what the mechanism involved it wasn't some sort of overall effect on aging. It was purely an effect preventing infection of the pharynx, right? It wasn't a sort of general aging effect, which would kind of perhaps argue for a sort of a non-general effect, but we were quite happy that it made sense. On the other hand, actually, a Nature editor came back and rather strenuously said we should sort of do some sort of retraction. Initially, I agreed, but then my co-authors, they just said, "Oh, for God's sake, I think there was a sense that it after all this time, it was, especially given the nature of the results, the FUDR dependence and the fact that it's just a sort of bacterial resistance effect." But yeah, it's a sort of interesting kind of final note, and at least it's possible to get things to make sense, to resolve these things. And the problem with FLIN2 was affecting the whole field. So yeah. In our defense.
Yeah, no, definitely highlights an important problem, right? Of using, are we actually using the same model organisms with the same sort of genotype and mutations and whatnot? So I'm curious, what does this mean for Sirtuin research in humans? Do you think it's still exciting and promising, or is it not really if
No, really.
begins? It's
I haven't stayed close to it. The involvement with sirtuins for me was very much because of projects that we were working on in the past, where we were trying to understand the mechanisms of dietary restriction. Mm-hmm. And I kind of came to the conclusion, I published this a long time ago, that the dietary restriction models in C. elegans are so hopelessly problematic that I felt I couldn't really work on them. There were too many problems, and I don't want to go into that. But my partner in this research consortium, which was Linda Partridge, she was kind of working more concertedly, because the fruit fly is a much better model for studying dietary restriction, I think, and they were getting very fruitful. They were getting lots of fantastic results. But the question was, maybe the answer had already been found, which was that, sirtuins essentially explain the effects of dietary restriction. But during the noughties, there was a whole series of problems with reproducibility of results, where results that were coming from a small group of labs who were incidentally linked to a company, where there were financial interests, were producing results that just everything was showing that the sirtuins were mediating the effects of dietary restriction on lifespan. So really, in a way, my particular interest was on that, and I think that certainly a lot of that work turned out to be it was. I have to be frank. It seemed to me like a nuisance within the field, the way that there were just a continuous production of things that were difficult to reproduce, and the impression that one gained was rather unsavory, that there was something unsavory happening there. Perhaps the possibility of conflict of interest, came to my mind, and I think to many people about it. There's a lot of discussion. There were articles, in Science and Nature about problems with sirtuins. Mm-hmm. But on the other hand, I mean, the discovery of sirtuins originally, their role in yeast aging, I mean, it led consequently to fantastic, rich biology. And I mean, the sirtuins are highly pleiotropic, involved in general ways in gene regulation and so on. I mean, it's a large field of interest. It's just a pity that there was this period where there just seemed to be, a veil of tears of things not being reproducible, endless quarrels and scandals. And I mean, it was terrible. It was a terrible area. One of my postdocs went, when I couldn't go to a conference in Cold Spring Harbor, I can't remember why. I mean, I'd given talks about the problems with the work. He went there and he said he came back, he felt like he'd been beaten up because he was presenting results that were not consistent with the sort of, the sort of accepted view of their importance. So it was quite nasty, the whole area.
Wow. Yeah. I was talking to Matt Kibelen, who said he was also involved in the discovery of sirtuins, but he said that, when so many labs showed that you can actually reproduce the results, he became less in favor of them, that perhaps it's not actually that interesting, and we should be honest about that.
I mean, in my defense, in terms of the Flynn 2 work, I mean, at least I corrected the work. I went and corrected it as best I could. And that certainly was very much not the pattern with the Sirtuin work in the noughties. And as I say, I haven't really followed it very much since then. I sort of run a mile from this topic, really.
Okay, so another
work in areas where there are good, useful findings and reproducible findings. Right? Where you get certain areas, they get fouled up with bad work, and it essentially tangles the work up.
Right. Oh, 100%. So another old paper of yours, which talks about how metformin alters the gut microbiome and also methionine metabolism. But it sounds like it depended on the E. coli strain and what the sensitivity to metformin or glucose concentration was. I'm curious, like, what did your work show in terms of how is metformin affecting the gut microbiome? And as a second part to the question, I think there was a recent study that came out that showed that metformin didn't really have beneficial effects. I don't know if you followed that and if you have any thoughts on that.
What paper is that? I honestly, to be honest, that the work on the microbiome, I had a period when I was kind of interested in the microbiome. And then after the metformin paper, that work was kind of in a way taken away from the lab. I mean, a lot of that work, a lot of the beautiful aspects of that work are not from me; they're from Felipe Cabrera, who really took that work and it's done beautiful work since. And I mean, I think that the thing that one of the things that I thought was really interesting about that work was the way that it raised the question of how host, drug, and microbiome interact, and how actually a critical part of understanding drug function is how it's biotransformed by the microbiome, and how the drug also affects the microbiome. So it's a complex system, and actually, C. elegans, the system where you have C. elegans and then you have E. coli, and then you have a drug is a fantastic system, a reductionist system where you can simplify. You have something that is manageable to study, where the real microbiome, like in mammals, is incredibly complex. But in C. elegans, you have a single bacterial species, and then you can alter the genotype of the bacteria in order to be able to understand interactions between drug, bacterium, and host. And there was a beautiful study that Felipe did after he set his own lab up, looking at floxuridine, which is used as a treatment for colon cancer. Mm-hmm. So you could really see how interactions between the bacteria and the drug were relevant to understanding how it works in preventing colon cancer. So I think it's a very, very useful model.
I see. Okay, interesting. Lastly, I think we discussed a little bit already on your critique of the hallmarks of aging. And one point that came up again and again was that, so far aging has just been viewed as molecular damage. But of course, we spoke at length about the programmatic theory of aging. But another point you make in that paper, a review, is that we stay away from the hallmarks of aging are biased away from things like immunosenescence or some of the other markers that are closer to the medical field. Maybe you can touch upon that point a little bit more.
Yeah. So first, let me say that when I originally read the Hallmarks of Aging lesson that first came out, my initial sense was, God, this is really useful, very useful, because the aging field gets so dispersed, that it gets so big that you rarely get things that are really kind of in a way covering the whole field. Mm-hmm. And it did it really well. It managed to cover in a way that was unusual a very wide range of different topics. But then it's sort of towards the end, it goes into this hallmarks. The whole hallmarks thing itself is a little bit strange. It was strange to me. So I had very mixed feelings about it. But I thought, well, the hallmarks, I just never mind that. It's a useful review. But then to my surprise, the more I'd go over the years, I'd go to meetings, more and more people would show this hallmarks diagram, and they would sort of refer to it as if it was some sort of central paradigm, as if the hallmarks thing really meant something. And it doesn't really. It's sort of, it's kind of odd. And I started wondering about why, why has the hallmarks become so prominent in the way that it has. I mean, maybe this sounds a bit like psychoanalysis or something, but I mean, my interpretation is that in a way, the kind of core paradigm that existed in the 90s, you know, with the damage maintenance paradigm, the Ross theory, the disposable soma, the allocation theory, and all of these sort of things, it's sort of fallen apart. And in a way, the field is more kind of fragmented than ever. And it's very difficult for a field as a community of people to function without having a core paradigm. So in a way, the Hallmarks kind of functions like a paradigm, but it actually isn't an explanatory paradigm in the proper sense. It's not like the periodic table or something. And it's certainly not like the Hallmarks of Cancer, which it's based on. The Hallmarks of Cancer is a fantastic paradigm. It's an enormously powerful, you know, explanatory power and also ability to guide research. So my sort of looking at the Hallmarks thing itself, especially towards the end, there's a kind of a flow chart that sort of shows the relationship between different hallmarks. Is that the flow chart at the end was still very, very much flavored with damage maintenance paradigm. It was almost like it didn't want to say that the damage maintenance paradigm was still the paradigm, and yet it was like there as almost in an almost ghostly form, you know. So it was almost as if it was damage maintenance paradigm by the back door. It was sort of smuggled in a funny way. And one of the things that's very different about the programmatic theory, and this goes back not only from Misha Blagosklony, but also Vladimir Dillman, they emphasize very strongly that actually, you know, aging is a process of accumulation. It is a process of multiple diseases, and that if you study aging, you're studying disease. And actually, this is a critique from, for example, from Misha and from Dillman, that the damage sort of theory kind of tends to focus attention on events within the cell. So you're looking at molecular events. You're looking at it's a molecular explanation, damage, and so on, mitochondrial biology and this sort of thing. That's where you're looking, and that's where you find an explanation for aging. And then from that, you have the cells become damaged or deranged or whatever, and then that sort of leads up somehow to different diseases. But the diseases themselves are kind of not very interesting. You know, they're very complicated. Whereas the programmatic theory really sees the diseases themselves as developmental events. That's really where the meaningful pathogenetic processes are happening. Mm-hmm. And I think one of the problems with the damage maintenance paradigm has been, because it focuses on the subcellular, it actually tends to not talk much about diseases of aging. And it's striking how that the Hallmarks is very consistent with that. And the programmatic theory, I mean, if you look at for example, the review that you mentioned about hyperfunction theory, it's much, much more oriented to looking at disease. And it also is critical of sometimes a distinction which is even made between aging and disease, which is an old medical, I think partly from an old medical tradition of sort of calling some of the deteriorative age changes normal, which I think is in a way a pragmatic practice for doctors dealing with elderly patients, just to not make them feel you sort of say, well, your eyesight's going, you're having problems peeing, and you've got some pains in your joints and weakness and so on. But for your age, you're doing really well. You're probably better than normal. That's just normal aging. It's nothing to worry about. So this is this is good practice, But from the biological point of view, the idea of sort of normal senescence, like sort of normal biological processes of senescence, in the sense that they're not pathological, but let's say non-pathological senescence, is a nonsense, really. So I think that in a way, yeah, that's sort of a part of the critique of the Hallmarks is that it's part of the sort of tendency to neglect, to not focus on diseases of aging, but focus on sort of some central mechanisms, particularly subcellular mechanisms. This is part of the theory that actually both Blagosconi and de Magalhães both made this point, that that a lot of the problems that emerge during aging are happening at the level of not of cellular homeostasis, not of cellular health, if you like, but tissue homeostasis and organ homeostasis and sort of higher physiological homeostasis. So a lot of the cells in later life, the cells are fine. Mm-hmm. It's more tissue homeostasis, particularly, that goes awry because of programmatic problems.
Interesting. Wow, fascinating. I'll actually be having Joao Pedro on the podcast as well, so look forward to talking more about it.
That will be, he's a very. Some people, they just keep having original creative insight. I mean, I think part of it is because he reads very widely, and that goes also for Misha Blagosconi. They read very widely. Misha also very much reads a lot of the medical literature, which I think biogerontologists tend not to look at so much.
Right, and that also explains, right, like maybe why he's thinking about why aren't we really thinking about the disease of aging as part of the aging process, as opposed to very distinct.
As a clinician, as of course was Vladimir Dilman.
Yeah, fantastic. If we have maybe 2 more minutes, the last thing I wanted to discuss was, you briefly mentioned this, the 2 causes of death in C. elegans, and it turns out it's the swollen pharynx or the atrophied pharynx, which largely is caused by just bacterial infection. I found it, I think in your paper you mentioned that, well, we could give them some sort of antibiotic and remove this infection and maybe be able to better study the intrinsic mechanisms of aging. But you actually end up saying, no, this is really telling, because the damage that's happening because of the swollen pharynx later integrates with the aging damage and sort of explains better the causes of aging. I think if you could explain that, because my thinking was that why don't we give them this antibiotic? I mean, humans take all these vaccines and antibiotics, and so maybe that's a better model.
It's a great question. It's a very tricky one. It's a very tricky and complex topic. So, for example, one of the preoccupations of people working on mouse aging is that in some cases, if you're looking at intervention to see if it extends lifespan, and then the control lifespan of the mice is very short, you say, well, the mice aren't dying of aging; they're dying of something else. So you're not necessarily affecting the aging process. So for a long time, this was sort of axiomatic in model organism biogerontology, that you're always trying to remove the extrinsic causes of the things that extrinsic factors that shorten lifespan. And there's some sense to that, and in fact, I mean, for example, you mentioned Matt Kaeberlein. I mean, I don't know if he still does, but he used to do all of his work on E. coli that was essentially irradiated with ultraviolet bacteria, so that it wouldn't infect the worms, to essentially take that out. But in fact, right in the paper, this was a point of argument. Does this mean that in fact, looking at what we're looking at with the 2 forms of death, big P and small p and so on, that we're just looking at infection, we're not looking at aging at all, and what we're looking at is uninformative with respect to aging? And I would argue not, and I can make I think a strong case for that, particularly because the multifactorial model that I just described to you, the sort of Dilman plus multifactorial model, actually I was able to sort of realize, to understand it, because it resembled an earlier model that we developed, Yuan Zhao and I, and Anne Gilliatt, who also worked on that project, which is a model for how aging is working, which is that, so what, this is a bit complicated, but so what we think happens with these big P deaths. So big P death, you get this swollen pharynx full of bacteria, and the worms die kind of prematurely, they die earlier. So what actually happens is very interesting. The young worms, they feed at a very, very high rate using this structure, the pharynx. And what happens is that the the cuticle of the pharynx experiences mechanical senescence. You get wear and tear, mechanical wear and tear on the pharyngeal cuticle. And what happens then is that bacteria invade the pharynx. Mm-hmm. But then you don't get an infection. What happens is that the damage, the pumping rate of the pharynx goes down, and the wounds in the pharynx heal up. And you can see scar tissue in the pharynx using electron microscopy. And what you get, and you can see this by marking the bacteria with a fluorescent reporter, you can see these little puncta inside the pharynx. And if you look under the electron microscope, you can see that they're actually membrane bound. So they're a little bit like granulomas in tuberculosis. The bacteria is contained. And so then for about a week or so, nothing happens in the pharynx. The bacteria are contained in these granulomas. And then when the major senescence process kicks in, they come out like the Trojans in the Trojan horse. They come out, they infect the pharynx, and they destroy the pharynx. Mm-hmm. So this provides a way of thinking about this. This is the two-factor model. The disruption is particularly the infection of the bacteria, but in the young worm, the infection is contained, right? But in the old worm, then it comes out, and you see this pattern where disruption is contained in the youthful organism, and then when senescence appears, it essentially it is unmasked, or to use the medical term, it shows recrudescence. So, for example, in old people, children often get chickenpox, and the immune system clears the chickenpox, but you actually often have some latent chickenpox virus. And in later life, as the immune system changes, the chickenpox virus can re-emerge in the form of shingles, which is a far more painful and unpleasant condition. And that's an example of where you have a disruption which is contained, and then it emerges in later life. And actually, this pattern where disruptions are contained and then emerge in later life, you see again and again in aging-related diseases. So I think, in a way, that general rule that we worked out to explain, it's in the supplement of that big P, small p paper, is actually a tiny version of Dillman's multifactorial model. So I think it goes to show that, in a way, to understand how aging-related diseases emerge, how they develop as multifactorial diseases, it's helpful. It doesn't hurt to have disruptions there that might reduce lifespan. So if we used UV irradiated bacteria, we wouldn't have learned that. It's a beautiful question.
Yeah, no, that's exactly what I was getting at. That some of the disruption you're saying, even in humans, it might happen when you're younger, but because we're youthful, it's contained.
Yeah.
But then as we get older, say the programmatic theory also kicks in, including the damage.
That's actually what looking at osteoarthritis, because in osteoarthritis you get injuries to joints when you're younger. You get a football injury to your knee, and then it's fine, and then you get into your mid-60s, and suddenly that knee develops osteoarthritis. It's like I put it. It's like a loaded gun with a trigger. It's like a gun with a trigger. So it's rather like you have the gun and it's not loaded. You pull the trigger, which is the injury to the knee. Nothing happens. When you get to 60, then the gun is loaded. And then you pull the trigger, and then you get your disease. You get the osteoarthritis. Yeah, and that's so common of human experience. I think even, say, marathon runners, they say that they'd be able to run miles when they're younger and their knee is fine, but then suddenly it starts hurting when they're much older, and it's impossible for them to run anymore. It's a recurrent pattern with lots of aging-related diseases. Fascinating. All right. I think we've covered a lot and taken up a lot of your time, but is there anything else that maybe we haven't discussed you wanted to talk about? Not really. I mean, covered a lot of ground. Good to talk to you, and it's really nice that, it's nice because this stuff it's available on YouTube. It helps people to have a chance to know what's going on in the field. Hi again, everyone. I hope you enjoyed the episode and learned from it. If so, please show some support by subscribing to my YouTube channel, Live Longer World, liking the video, sharing the podcast, and leaving a review on Apple Podcasts.
To be notified of upcoming podcast releases, sign up on my website at livelongerworld.com. Follow me on Twitter at livelongerworld and Instagram at longevityfuture. Thanks for listening, stay in good health, and I'll catch you soon.